Okay, I think we can start now. Welcome to everybody also on YouTube. The title of today's talk is Current Developments in the Global Genome Commons and Challenges for Service Provision in NFDI. The underlying topic is really very relevant, but before we start on the content, I would like to introduce the two speakers of today's talk to you. Both are highly involved not only in NFDI, but also beyond in many different groups. Our first presenter is Professor Dr. Konrad Förstner, who's the speaker of the consortium NFDI for microbiota. And he's also a joint professor for data and information literacy at the Technische Hochschule Köln and ZB Med. He is the founding member of the working group Open Science at the Open Knowledge Foundation Germany. And he represents the German Rectors Conference in the working group Digital Tools, Software and Services in the Priority Initiative Digital Information. And this is only just to mention some fields of activity Konrad Förstner is doing. Professor Dr. Jörg Overmann today represents the NFDI, the consortium NFDI for biodiversity. And he is also the director or one of the directors of the Leibniz Institut Deutsche Sammlung von Mikroorganismen and Zellkulturen in Braunschweig. And he's also a full professor of microbiology at the Braunschweig University of Technology. Besides this, he is a member of the Permanent Senate Commission on Fundamental Issues of Biological Diversity of the German Research Foundation and member of the Council of Scientists at the Human Frontier Science Program Organization. And he also serves in several supervisory boards and scientific advisory boards of national and international institutions. What is today's talk about? It's about digital sequence information, DSI. And this is a term which is used in the context of certain international political bodies, in particular the Convention on Biological Diversity. Digital sequence information refers to all genetic resource data. These data are stored in many international and interlinked databases and are available and also very important for the research community. At the moment, there's a worldwide discussion to restrict access to these data. Perhaps some of you also already got familiar with the word Nagoya Protocol, which is an important term in this context. This discussion or the restriction of access can have devastating effects on international research and which can also collide with the interests and aims of NFDI. And in this in today's InfraTalk, this will be discussed mainly in the context of the Life Sciences Consortia NFDI for Biodiversity, for Microbiota, Data Plant, and the Consortium Initiative Fair Agro. I'm really looking forward to this talk, and I'm handing over to Konrad Förstner now.
Yeah, thank you. Can you hear me? Does this work out?
Perfectly.
Great. Thank you so much also for the very kind introduction, and it's a pleasure to discuss this topic today with you. I will get started and then later on Jörg Overmann will take over. The short description already mentioned this. We will talk about something which is called DSI, digital sequence information, which is a rather broad, or can be broadly interpreted, and we will see what this all means. Why this has an impact on us here as the life science NFDI consortia, it can go far beyond this. And this is again the question of interpretation pretty closely. Also the, let's say, consortia of the medical field could be addressed, but this goes, as said, much beyond this, and Jörg Overmann will shed light on these potential risks. in a way, you might be familiar with a few parts of this topic already. Jörg Overmann gave last year already a talk in this direction, and this kind of an update or an extension in this direction. So we would basically stick to this topic and push it further, because we see here, huge risk to basically everything that we want to do with NFDI in general, and we will hopefully be able to show you this during the talk. At first, what is this digital sequence information? Very precisely speaking, this would be actually only DNA, RNA, and protein. So basically biomolecules, if you get to their sequence, this is basically the classical sequence information. But this can be and is much broader discussed and interpreted today. So basically you have the subsidiary information like the genetic resources, but also metabolites, so small molecules inside of cells. And you see this is going far beyond the normal definition that a biologist would maybe take or a bioinformatician would take. But in this political context, this is extended, and here lies a large risk, because we could actually extend this to many other types of data that is of high interest for the research field or for the research and researchers in general, and due to this to humankind. Maybe a small step back to biology. If you're not coming from the field, you have DNA. So this blueprint of life, as it's often called, this is transcribed into something which is called mRNA or other RNAs, but mRNA is, let's say, the most prominent one. And this is then translated into protein, the actual players in the cell, although some RNA biologists like myself would maybe argue that the RNA is also of higher interest, but this is not the topic for today. So basically, this is the original interpretation or the original or the origin of that. While, as mentioned, this goes far beyond that. Just that you have a rough image in mind where this all started, but that you can also see where this is going if you also use this term also for a different type of data. Today, there are already many resources that take care of all this kind of information, and this is great that in recent decades already, these kind of resources were established internationally. The community could not work without that. This is pretty clear. You could not do any biological research without relying or working with these kind of resources. And that is already great. And I think the life sciences have here quite a pilot or pioneering role in the scientific community. Still, there are a lot of gaps. This is why the NFDIs in life sciences exist, and they want to clearly improve this. This is, as said, very international, and the most important thing, it's also very connected. So if you want to check a gene, for example, in one database, you find a link to the protein in another one. So it's very hard to dissect this, and the actual value lies in the connection of these resources, so that you can actually go from one entity to another and see the broader picture. Without this knowledge is, I wouldn't say worthless, but it loses dramatic on value. And here to point to two resources that are actually part of NFDI for biodiversity, Brenda and Silva, are definitely one part of this network and are embedded in this international set of resources. So this is key, and we will get back to this later on, because this is kind of the key part, that you cannot simply separate a single resource and continue working with this. The value lies in the connection between these different resources, and they are a fundamental resource for the research community. In NFDI, in all of them, in all of the NFDI consortia that exist, there's clearly a certain value chain that we are aiming for. So we start by the experiments, we do the data generation and take care that this is all done properly, including metadata and so on. We provide analytical workflows that help to analyze the data, but also to publish the data later on and to bring this into publications, and later on to include and add structured data. Basically, these resources that we talked before about are kind of distillates out of these publications, out of the data, in order to make it accessible. So this is something shared between all our consortia, and this common aim is fundamental to all of us. And this is also where NFDI can still really make a change, because there are currently friction in the system, and improving this will tremendously improve the way researchers work, that you actually have kind of automated ways of bringing or starting from the experiment down to the publication, that this all becomes kind of frictionless and seamless. This is what we are all aiming for. And there are very huge gaps partially, and we try to solve and fill those gaps. I start here with NFDI for microbiota, just as a first example which this basically started last year. And just to very briefly mention what we are aiming for is that we bring this kind of value chain to the research on bacteria, viruses, archaea, protists, parasites, and fungi. So everything that is pretty small and living out there. And there are many questions that still are open, and where actually good data management and analytics can help to answer questions. So in general, we want to look at the different molecular entities, the different species, but also the molecules in there, and more importantly, the interaction between them. And this is basically what we try to enhance, or where we try to help researchers to get these questions answered in a much quicker way. So for example, and here comes also the connection to other consortia into play, which bacteria, fungi, or viruses are relevant for a crop plant. Right? So the plants themselves, they cannot live in isolation, but actually the microbiome is crucial for their survival and also for the proper growth. And knowing what is in there and what is supporting a plant is crucial also for the agriculture and everything on top of this. And you are clearly aware that the microbiome in and on our body are relevant to our own health, and basically that whenever we figure out this connection, we can also work and engineer in a better direction. It's also clear that microbes can make us sick, and there can be bacteria, there can be viruses, that can be fungi. So what is actually making us sick, and how is the interaction happening here? But also to heal things. So for example, antibiotics are produced by different microbial species, and they act against other bacterial species or microbial species. And which one are those? So these are things that we would like to actually work on for those. These digital sequence information are crucial. In NFDI for biodiversity, the spectrum, in my opinion, is even broader. Yeah, there you have actually very heterogeneous data in terms of the organisms, the species that you look at. You have to look at ecosystem data, for example. You have different experiments. You have, but also you have molecular data, and you need to link this to climate data. So this is a very broad spectrum of data that comes here together, and that needs to be connected in order to really get value out of the whole system. You cannot look separately at one entity or at one of these databases in order to really see the global picture, and this is key here. And for this, NFDI for biodiversity is already rather far and has basically established, or is working on the establishment of this system here, the Research Data Commons. And what is the key part here is that you have transition of data through these layers, and there's an interplay between these different layers, and that again here the separation between different entities and databases is artificial, and it is crucial that you can get from one database to the other, and can by that create actually a knowledge by itself. But there are also, let's say, joint images or joint endeavors. If we look, for example, at plants, and this is here again from Data Plant for Agro and NFDI for Biodiversity. If you want to improve crop plants, you need a lot of different information linked together. And the aim for this could be that you want to have higher yields, or that you have adaptations to stress due to climate change. Again, you need different information in here, that you need resilience to different pathogens. A lot of the harvest today is basically getting destroyed by, again, microbial species, for example. And in the end, you would like to do biotechnology and basically change the environment or the genetic material of plants in order to adapt to that. And the issue is that this needs a long time. Plant breeding is basically something that really requires a lot of time resources. And due to this, it's key that we have the best knowledge in order to, engineer in the right direction. For this, stable identifiers, if you think in terms of data are required, that even over a long time period that data can be tracked and that this can be properly referred to this. So this is kind of addressed by these different consortia. And this is something that we getting to later is with those identifiers, you also have hopefully a clear legal situation that you can refer to your material, what you have at hand and where this originally comes from. There is a lot of development inside of these consortia, and here's just one example which I find very impressive as not being from those consortia. The sheer diversity of plants is basically asking for very complex models to precisely describe the specimen and the basic material in the plant sample. Yeah? So you can have a plant, but you can take different samples of this. You can take the seed, you can take something from the leaves, you can take something from the stem, and then you describe this phenotypically with a microscope, doing with sequencing. And then you basically have to model this and put this stuff into relation. And this clearly, as you can see, just is quite a challenge, and also is not done in isolation, but the opposite. This has to be always done in connection to other databases. And this connection, again, leads to the extra value, to the extra knowledge that is required to do efficient research. And here's basically the model. It needs to be extended in order to describe the samples properly, and this means the biological material, but also the source itself, so where this is basically coming from. And then the data is enriched by taxonomy, but also by variety, by geolocation, and by climate data, and all that together. You see how all this things cannot be separated, but the key part is the synergistic effects if you bring the data together. So if you have proper identifiers that are linking or can be used to link different databases, and basically have a joint look at the data and make the right connections here, and also make it able to query them. So fair data. So in principle, these stable identifiers are kind of key in order to follow digital sequence information along the data life cycle. And here are a lot of developments ongoing, especially in these different consortia. And the aim is basically to support the researchers in this whole part of the research life cycle. And as said, several new models need to be developed here, and this is ongoing work inside of the different consortia. This is all just necessary for you to understand. So the key part is the connectivity between the databases. The key part is that we really gain a lot of value simply by the connection of the different resources. And that is basically now the place where I can hand over to Jörg Overmann in order to, well, tell us what are the issues with this today.
Yes, I'll take over. And after showing the entire data ecosystem we are used to and we are working with, let me first outline how we got in the present situation, what stimulated the discussions and what the problems are. And for that, we have to go back to the United Nations Convention on Biological Diversity that was actually starting in 1993. And which is an international instrument to address the issues of biological diversity, use, protection, and even green growth, where it started. It turns out that we now have a situation where which is comparable to the negotiations with respect to climate change. And you all know that there was a Paris Agreement, which was considered a certain breakthrough and which took long nights to negotiate and to actually agree upon. And we have a similar situation now, which is called the Global Biodiversity Framework, which has 21 targets and 10 milestones, and which is currently under intensive debate and lays the foundation for a better protection of biodiversity and halting biodiversity loss, and as such the basis of our living. And the problem that has emerged is that in this connection, digital sequence information plays a major role. And the issue is that there is not only the desire of many countries to set up this global biodiversity framework, but there is also the need seen to deal with digital sequence information as a source of income. And now these 2 points, which at first glance seem to be separated, are negotiated together. And on the next slide, that is a little bit more detailed. The reason why DSI has such a prominent role in this essential negotiation of the global biodiversity framework is that some countries and some protagonists argue that DSI offers a loophole to avoid the obligations for access and benefit sharing. In other words, it is envisioned that sequences from unique organisms in, say, the rainforest or in some special environments can leave the country of origin and can be used without being obliged to access and benefit sharing with the country of origin outside. And therefore, these many countries really try to find ways of controlling digital sequence information. And they argue if there is no access and benefit sharing regime for digital sequence information during the current conference or the conference of the Party 15, which is going to probably take place somewhere end of August or a little bit later, then there will also not be a global biodiversity framework. What is meant by controlling for digital sequence information can be shown by several short phrases which we put in here, which have been actually stated during the recent negotiations in Geneva. For instance, that there should be an intrinsic link between the DSI and the genetic resources, that there is the sovereign rights of countries over DSI. There must be links between DSI and the country of origin. There must be traceability, and open access is not free access. So all these statements point very clearly to the direction to control the access to digital sequence information originating from certain countries. We have to also consider that there are already bilateral measures. In other words, there are regulations for dealing with some countries, in over 30 countries actually, to get access to digital sequence information. So for some of these countries, for more than 30 countries, you will not be able to freely access digital sequence information from any genetic resource from that particular country. Another complication is that digital sequence information, or the definition of digital sequence information, is ambiguous. You have seen already the introductory slide of Konrad, and I'll show you how far that actually can go. And there is presently no real consensus. Oops, yeah, the next one. And Konrad Förstner has already pointed out that in our data environment, we know and we experience day to day that there are strong links and interactions between different types of data, and the strength of the links between the different core data resources of Elixir that is shown here point to the many different data types which are exchanged basically day to day. And this is not a unilateral or a one-way road, but as we know, it's going back and forth, and it enriches data in databases by information from outside. On the next slide, this is a little bit more exemplified for a specific case from Brazil. This has been a study by Amber Scholz of our DSMZ here in my group, who have analyzed which sequences are actually used in publications on biodiversity or in the broader sense. And it turns out, if you, for instance, take Brazil as an example, that there are actually 108 countries, or I should say authors from 108 countries who actually use data from Brazil. And on average, you see the numbers here, there are 44 sequences which are used per publication. There's a broad range, of course, of these numbers. Now, if we turn around and ask how many data from how many countries are used by researchers in Brazil, you see that this is even higher. In this case, Brazilian researchers are dependent on 141 countries and their digital sequence information. You see the link there, how this was generated, but it clearly shows in just one example how in a country of the global south, researchers are as similarly as we are dependent on sequence information from outside and their availability. If you now look at the next slides, this is what would happen if digital sequence information would be under an ABS, an access and benefit sharing regime as defined by the Nagoya Protocol. And you can replace project by access to digital sequence information here. So currently what you need to do if you want to get access to bacteria, plants, animals, whatever in a country, you have to decide, is that a case for access and benefit sharing? In other words, is that country a party to the Nagoya Protocol, which most of the countries are? Then you have to identify the specific regulations and the authorities you have to contact in that particular country. You have to negotiate a prior informed consent with the authorities, the proper authorities in that country. And if that is signed, you then have to negotiate the mutually agreed terms, MAT. These are all technical terms of documents which you have to acquire before you even can start of planning your sampling. And if you then want to get the sample out of the country, you need usually a material transfer agreement, and then only research starts. Envision that this left part of the graph, this left branch, is what you need to do for each and every single data point from different countries. So if you're using data from 108 countries, as we have seen in the Brazilian sample, for instance, you need to do that exactly that many times. And remember, the regulations are different. It becomes very clear that the current construct, how to deal or how to ensure access and benefit sharing under the Nagoya Protocol for physical resources, is not at all applicable to what we know and how the data environment we have and how this data environment actually works. Let's go to the next slide. It is even worse, and this is from the same publication, and it plots how many data are provided compared to how many data are used in different countries. And the countries of the global south, developed countries, the G77 are marked in green, rectangles here, the OECD, so the industrialized countries in red, and the BRICS countries, that would be Brazil, Russia, India, South Africa, and China, they're plotted in blue. And if you had an equal sharing, that means a country provides as many digital sequence information, that is sequences in that quantification, in 118,000 publications, that is, as it uses, you would be on the dashed line. And what you see very clearly is you have an area where scientists in countries are actually using more than that country is providing to the global data pool. So whenever you're below the dashed line, you're using more DSI than you're providing to the global data system. If you're above, like the yellow dots, then you're providing more than you're actually using from other countries. In other words, the G77, many of the G77 countries are very much dependent on the access to digital sequence information from outside, which means that wherever this data utilization exceeds data provision, like for many of these countries, they will be heavily cut off from the data they need so desperately for further development of the competencies, of the infrastructures, of the science. So a regulation as is envisioned under the Nagoya Protocol would actually, and this is by inference from these very data, would hit those countries the hardest, which is exactly the opposite of what is intended, of course. So on the next slide, I want to report on another complication. It has been already shown in the second slide or so what digital sequence information is in our perception. But the AHTEC technical expert group, which informs the CBD Secretariat on scientific facts and so on, has discussed that within DSI on genetic resources, there could also be data on ecological relationships, abiotic factors of the environment, even behavioral data of animals or phenotypic data. You can see that this extends far beyond anything we can envision if that is controlled. And I just gave you one example, and that actually expands, and I see that some representatives of other consortia are here as well, which are not dealing with sequence information, I guess. It extends all the way to the cultural realm. And so I think, and most scientists involved are convinced that this route is not the right way to support data sciences in developing countries, to foster international collaboration, to actually support these countries in developing their own bioeconomy, and to solve the issue of fair access and benefit sharing. Probably the opposite will be the case. Next slide, please. So we can conclude knowledge generations requires first a considerable investment, and we see that this investment is still going to be needed in many countries of the world for them to actually exploit the data better than before. We have seen sequence analysis or data analysis is multidirectional. So any digital sequence information behind a paywall, something like Netflix, or something similar, will become a shell format because it is exempt from the enrichment with other data from outside. And I've also shown you data which clearly show that the old stereotypes in this discussion are certainly wrong. The providers are not the developing countries and the users are not the industrialized countries. And this is a real issue in the discussions to make that more clear. Now, you might ask, what does it mean to us? Well, first of all, the NFDI has set out to foster data exchange, data analysis, harmonize this, so actually make big steps forward in the dealing with the data for the good and benefit of everybody. So there are some conclusions what we need to do, and that's on the next slide. We need to make sure that political decision makers are very clear that any paywall whatsoever will not work, using, for instance, the arguments I've been showing. We need to inform about the consequences. What happens if this heavy and very fruitful data exchange is simply cut off? There is already a large group of international researchers, and I should emphasize this is not only German, not only European, it's by far more than that. It's joining scientists, mostly biologists, but also oceanographers and so on, around the world from all the continents. I've given you the address here. You may join this network in an attempt to inform policymakers on this high level, on the scientific facts and the scientific consequences of what is currently being discussed. There is already an open letter. You will find that at the website, which you may sign if you want. It has been signed by many, many scientists around the world, among them from South America, from Brazil, you name it. I should emphasize that time is short. There will be, and that has been necessary because of the huge dispute between parties, there will be an intersessional meeting of the parties in Nairobi pretty soon. You see the date here. And there is, of course, the large conference, or the official conference of the parties 15 in Kunming, which has so far tentatively scheduled August 22. We are not entirely sure, but that will be the place where finally decisions will be made, and these decisions will affect not only our, but work worldwide with data dramatically if the wrong decisions are going to be taken. So it is, I think, very important that also NFDI takes measures and discusses strategies to enter the discussion on, and to make it very clear what is at stake and what the detrimental consequences would be if restricting data access and data use according to this model of the Nagoya Protocol, which would not work. And with that, thank you for your attention, and we both would be happy to answer any questions.
Thank you, Professor Förstner and Jörg Overmann, for your talk. I think it was again good insight and a deep insight in what the Nagoya Protocol is about, and into what this also means for interconnected databases. I think we are now reaching the point of discussion, which means that we would close down the YouTube live stream. And so